This is for the intestinal digestion activity A, digestion of starch. The semi-liquid produced during gastric digestion passes into the small intestine, stimulating the production of alkaline intestinal fluids. When the semi-liquid becomes alkaline, gastric digestion ceases and intestinal digestion begins. Pancreatic juice is secreted by the pancreas into the duodenum. This juice contains the enzyme amylase, which breaks down starch or breaks starch down into glucose, which can be absorbed into the body through the lining of the small intestine. So what we're going to do in this lab is we're actually going to test the optimal temperature for the breakdown of starch. So we're really testing what the optimal temperature is for the enzyme amylase. And we have the enzyme amylase in this liquid pancreatin here. Liquid pancreatin has the enzyme amylase in it, and that's what we're going to use to digest the starch. And we're going to test it at 3 different temperatures. We're going to test it at room temperature, we're going to test it at 40 degrees, a 40 degrees water bath, and we're also going to test a 75 degrees water bath, and we're going to see which temperature is optimal for the enzyme amylase, which is present in the pancreatic juice. So we're going to do 5 mils of pancreatic juice into each test tube, along with 5 mils of starch into each test tube. Test tube 1 will remain at room temperature. Test tube 2 will go in the 40 degree water bath. And test tube 3 will go in the 75 degree water bath. They're going to sit in there for 30 minutes, and after that time we'll put a chemical indicator called iodine, potassium iodide, I just call it iodine, into each of the containers to see if starch is still present. So if starch is present, you'll see a dark blackish blue color, and if it's not present, then you should see kind of like a tea color, something kind of light brown, transparent color. So we're expecting to have some breakdown of the starch in each container, but whether there's more breakdown in one over the other as a result of the optimal temperature is what we're really testing for.